Good afternoon, everyone. May I have your attention, please? That usually takes a little while. It's my pleasure to welcome Eric Trautmann back to Thayer School. Eric is currently a Ph.D. candidate in neurosciences at Stanford University in the Neural Prosthetic Systems Lab led by Dr. Krishna Shenoy. He obtained his bachelor's and master's degrees from Thayer in 2008 and 2009, working to design and build the autonomous Yeti robot for surveying Antarctic ice sheets as part of his 8990 project, and subsequently that was his research for his MS project as well. Upon graduation, Eric worked for 2 years at Physical Sciences, Inc., where he designed a quad-rotor helicopter drone and developed sensor fusion algorithms for application in defense. He began his Ph.D. at Stanford in 2011, applying his background in electrical engineering, mechatronics, and machine learning to develop novel tools and analysis methods for understanding population dynamics of neural activity, focusing on the primate motor system. Please welcome our speaker.
Thank you very much, Alex. It's a really special opportunity for me to be back home here at Thayer after just a few years of being away. The way that I got interested in neuroscience research and neural prosthetics in particular was in 2008 when John Cunningham, a former student of Thayer, came back and gave a very similar lecture. And through a series of somewhat serendipitous events, I somewhat accidentally ended up working in the same exact lab that he came from. He's now off at Columbia as a professor there. So one of my major goals of this talk is to encourage students with a background in diverse areas of engineering to consider areas of biomedical research and in particular neuroscience as fields where you can take that background and apply it to very specific problems where those skills are highly sought after and sometimes richly rewarded. So what I'll be talking about today are brain-machine interfaces and neural prosthetics, and using this as a motivation for developing new tools to study neural activity in vivo in awake and behaving primates. And so there's kind of two parts to this talk. The first part will be discussing the neural prosthetics, the current state of the art, where we are, where we're going. And then I'll tell you a little bit about our work to develop these novel tools for imaging the population of neurons at single cellular resolution, where we hope to record from many hundreds and potentially thousands of neurons simultaneously in awake monkeys that are playing video games. So, and use that to learn about the motor system in ways that will feed back and help us to design and build the next generation of prosthetic devices. So I'll start out with some relatively sobering statistics. There are actually several million individuals in the U.S. who are affected by some form of paralysis, whether due to neurological disease, spinal cord injury, or stroke, or several other factors. So not all of these are individuals with full tetraplegia, but many suffer from some form of motor deficits as a result. And those with full tetraplegia number in the low hundreds of thousands of individuals in the U.S. And obviously this is a global problem. So to put a face on this, many of you are familiar with Christopher Reeve, who suffered a spinal cord injury while he was thrown from a horse, and since that day has been unable to move any muscles below his neck. And despite Christopher Reeves, formerly Superman, being a man of considerable means, we have very few clinically available technologies that we can use to improve his quality of life. So there's no commercial products which we can give him to restore his ability to move naturally through the world and communicate easily. And there are many, many, many patients in his position. So what we'd like to do, the the overall dream is to give him a system to restore the basic motor functionality and potentially communications ability by recording neurons directly from his brain, bypassing the injury site, and using that to control a prosthetic device. So, in a normal, healthy individual, there's a region of the brain termed the motor cortex, which sends the appropriate control signals to your muscle, to approximately 45 muscles in your wrist, arm, and shoulder. And these go through the spinal cord out to your muscle. Now, in the case of neurological disease or stroke or spinal cord injury, these signals are unable to actually get to the muscles, but the brain is still operating in its normal way, still generating the appropriate control signals. So in neural prosthetics, what we'd like to do is read those signals directly from the brain, send them through a decoder where we infer the intended action that individual wants to make, and then use that to directly drive a prosthetic device. Now, this prosthetic device can be a cursor on a computer screen. It could be a robotic arm. In the future, we might see a full exoskeleton or some type of device which, or controlling a wheelchair or something like this. So we can think of this as being a data stream, which is the intended movements of an individual, and using that to control any sort of effector tool. So the outline of what I'll be talking about today, I'll tell you about how these current neural prosthetic systems function, how they operate, the general principles, and then highlight some challenges and limitations facing our current generation of prosthetics. So we'll then use this to motivate the need to study the fundamental underlying neuroscience in greater depth to understand how the brain is operating in healthy individuals. And then I'll tell you one of our recent efforts to do just that. We're developing a new tool called in vivo calcium imaging, where we hope to image in real time the activity of large populations of neurons at cellular resolution in monkeys. Now, this is a technique that has been established in fruit flies and in small rodents, and now we're hoping to scale this up and bring this to primates, where we can train very complex and rich behaviors, which will allow us to get very fine-grained access to understand how the motor system is operating. And it's our sincere hope that in doing so, we can take insights, learn from insights from what we learn about how the motor cortex is operating, and use that to then design prosthetic devices which are tailored to the specific functionality there. So, zooming in on the brain, the human brain is separated into different functional areas that are responsible for different aspects of how we interact with the world. There are certain areas of the brain in the back responsible for visual processing, and then the motor cortex is this bilateral strip on the back end of the frontal cortex, and it involves and these areas are principally responsible for controlling the muscles in your entire body. Now, there are other areas of the brain that are also heavily involved in motor control, but oftentimes those areas act through the motor cortex before sending projections out to the spinal cord and onto the muscles. So we tend to spend most of our time focusing on this region, but there are also very fruitful areas of study in the cerebellum and elsewhere. Now, back in the 1950s, several neuroscientists, Woolley and Penfield, did experiments with epilepsy patients where they had access to the cortex of these individuals who were undergoing neurosurgical procedures. And using microelectrodes, they put a tiny bit of current into the brain tissue. And what they discovered was that you can evoke direct movements in specific muscle groups when you stimulate in certain regions. And when they stimulated in some regions, they would evoke movements in certain muscles. And then when they moved the electrode over, they would evoke other movements in different sets of muscles. And this was the first time that anyone had shown in humans that functionality is highly localized. And what they were able to do is by doing very systematic and careful studies, you can build up a map of the entire motor system in the motor cortex here. And that map looks something like this. So you'll see that there's areas responsible for controlling the leg are sort of these medial areas, and we have arm and face, and then other areas represented elsewhere. So one feature that they noticed is that the areas that we think of as having very fine-grained and sensitive control and needing a lot of cortical processing take up larger swaths of the cortex. So the area that represents the hand in particular and the arm is very, very large, whereas we don't see large areas representing the back or other less significant areas. So this is actually great news for us in neural prosthetics. If we're interested in recording from neurons that are representing the motion of the arm, then we have a wide swath of cortex where we can target these electrodes. And it makes our requirements in terms of intra-op procedures, it makes it easier to target those electrodes when we're doing a neurosurgery. So how do we actually measure the neural activity in the motor cortex? So starting from the motor cortex, we have these silicon electrode arrays that are pictured here against the backdrop of a penny for scale. These are manufactured through standard silicon microfabrication techniques. They measure about 4 millimeters on a square. And then zooming into that, we can see about 100 small electrodes. Now this is still macroscopic. I mean, this bar here is about 2 millimeters. But this is the state-of-the-art technology. So we have 100 electrode arrays that are connected with a thin wire bundle to what's called a head stage, which digitizes the signals and then sends it on to the subsequent computers where we run our decode algorithms. And zooming in again on an individual electrode, we can each electrode is measuring the neural activity of the neurons just in the very vicinity of the tip. So it'll be surrounded by hundreds of neurons, but you're really only listening to the electrical activity of the neurons that are within 15 to 25 microns of the tip of the electrode. And sometimes you'll hear anywhere between 0 and 4 neurons on an individual channel, and this will give you potentially up to several hundred neurons to listen to. But as you can imagine, we can't individually target these electrodes. We just place this entire electrode on the surface of the cortex, and it's luck of the draw. You get what you get. Sometimes you may get 100 fantastic channels with a lot of very rich signals, and other times you may be less lucky and get fewer quality neurons. So this is one of the constraints and one of the key variables in terms of the surgical outcomes. So if we look at the electrical trace from one of these electrodes, we might see something like this, where we see voltage swings on the order of millivolts, and we'll see certain patterns of activity that might correspond to resting state or movement in one direction, and then what we'll see over here is these sharp vertical deflections which correspond to the neurons spiking. These are action potentials that you may have heard of, and these are the fundamental unit of currency of information in the brain. Neurons are all connected together and signal to each other by sending these spikes. So if we were to record from an individual here, as the individual is moving their arm to the left, you might see no activity at all on one particular channel, and then you might see a very different pattern of activity corresponding to a distinct movement. And so this is one neuron, and we will see very different and distinct patterns of activity on all of our different channels, and this is the fundamental basis of the decode. And I'll go into more depth in that in just a bit. I realize we actually would like to play some audio. Is that something that's easy to set up here? Let's see.
Associated with this guy, and then we need to crank this up.
But you might need to switch to the... Oh, it's okay. I can do it through this mic.
On your HDMI, you might have...
That's no problem. Here, what I'll do, I'll just hold this close to the speaker. There, so, yeah. So this is actually the audio associated with, these are recordings that were performed in a monkey reaching to targets on a touch screen in front of him. And you can see these distinct patterns of activity corresponding to these distinct movements. So how do we map this spiking activity, once we've recorded this, into a framework where we can use traditional mathematical tools that we're accustomed to in engineering? What we think of as being the important state variables represented in the neurons and the neural system in general is the firing rate of the neuron, how fast the neuron's spiking. Not each individual spike per se, but the underlying firing rate. And so once we create a smooth firing rate, we can then analyze those using our traditional tools. So here I'm showing you the reaching trajectories of a monkey's hand as he reaches to a number of different targets arranged around a circle. So these red lines are reaches to targets up and to the left, and the green lines are reaches to targets down and to the right. And we can look at the activity of a single cell as he's reaching. This is the firing rate of that cell corresponding to reaches to these different targets. And what we see is that distinct reaches have very different patterns of activity for this given cell. And each cell may look completely different, but they have these characteristic patterns which are tuned to the direction of the reach. So we can see that these patterns of wiggles are very distinct for these different movements. And this is the fundamental basis behind which we can perform a decode on the neural activity to infer the intended kinematics of a subject with paralysis as they're thinking about making a reach. We have these distinct neural patterns of activity, which we can then decode online and use that to direct the movement of the prosthetic device. So this was first successfully done in 2006 by Lee Hochberg and another group at Brown, who implanted the first tetraplegic patient with an electrode array. And subsequent work has made considerable progress in advancing the state of the art. And I'm going to show you a very quick video here. This is actually a clip from 60 Minutes, which will show you one of the patients controlling a robotic arm.
Plugged her brain into the computer, and this is what we saw.
I can move it up and straight down and left and right and diagonally. I can close it and open it, and I can go forward and back.
That is just the most astounding thing I've ever seen. Can we shake hands?
Sure.
No, really.
Yeah.
Like, come right over here?
Yes, you come over
Okay.
there. Grasp your hand there. There we go.
Oh, my goodness. Wow.
And I can do a fist bump if you'd like.
That's amazing. What are you doing, Jan? What's going on in your mind as you're moving this arm around? What are you thinking?
Okay, the best way to explain it is raise your arm.
Uh-huh.
Good. Now,
Right.
what did you think about when you did that?
Well, not very much. I do it all the time.
Exactly. It's automatic.
Is that hard work? Are you having to concentrate?
So this is quite encouraging. I mean, this is relatively high performance. You can see she's able to successfully move this arm in ways and is otherwise completely unable to interact with the rest of the world. But there are also some key limitations, and I'll show you one more video highlighting just a few of those limitations.
That will help with some of the things that Jan has trouble with.
Okay.
For example, sometimes when she looks right at an object, she can't grab it.
Okay, I'm going to take the cone away. Just go ahead and close it.
Oh, sure.
So as soon as
no
I take the
problem.
cone away, there's no problem. But as soon as I put the cone there, she can't do it.
Why
Yeah, well,
is still a mystery.
So this highlights one of the major challenges, which is that we can use the statistical properties of these neurons to decode the intended movements of the individual with paralysis. But we don't really have a deep understanding of the underlying motor physiology. We don't understand what the brain is trying to do in a healthy subject. And so it's very difficult to take those decoders and then address these limitations. We don't know why she's unable to grasp this. And then there's additional things. You'll notice that she was able to do most of these things, but it was not moving as quickly, as smoothly, as fluidly as you might move with a natural arm. And so as we think about scaling up to higher degree of freedom control prosthetic devices, and as we think about improving the performance and robustness, the ability to stop on target, the ability to make very fine control, we believe that it's going to be important to incorporate more information about the underlying motor physiology than we currently have. And so in order to do so, we'd like to develop new tools to scale up our ability to record from the brain and to ask different types of scientific questions. So there are three broad classes of challenges that make BMI systems not clinically viable right now. And the first broad class is that several straightforward real-world engineering challenges remain. So these are things like developing low-power, wireless, implantable electronic devices that are all things that we know how to do today. We can build iPhones. We can take what we learn from standard electrical engineering and then apply those to this problem domain and create these low-power, less implantable devices, wireless implants. So we also want to get rid of the computers. Currently, the decode algorithms are running in large workstations, and this needs to be something that is miniaturized and turned into a product. And then there's also straightforward questions like, how do we attach a robotic arm to the patient? We need better, faster, low-power robotics to do these. So these types of challenges are all things that sit firmly within the wheelhouse of everybody in this room. These are things that where there will be major opportunities in the mid and near term. So the second broad class of challenges facing current BMI are that there's several fundamental engineering and neuroscientific challenges. So these are, we want to improve the performance, as I mentioned earlier. We want to improve the precision, the speed, the stopping ability, and push this as close to natural motor control as we possibly can. Second, we need the robustness to improve. As you implant if you implant the current electrode arrays, you'll notice that the signal amplitude tends to roll off over the course of months or the first year or two. Now, we can still do reliable decode after 3.5 years. Our lab was one of the first to show that. But this is something where you need very repeatable long-term performance out to 5 or 10 years in order to justify doing an invasive neurosurgery such as this. Human neurosurgeons like to joke that it's minimally invasive because it's just touching the outer surface of the cortex, but you still are doing a neurosurgery. And then finally, we need new basic neuroscience knowledge. So this is, we want to understand how the brain prepares and controls movements in healthy individuals, and then we also want to understand how feedback is incorporated. So this is visual and proprioceptive sensory feedback. So the way our lab works, we use both primate and human subjects to address primarily the second two of those challenges that I highlighted. So on the primate side of the lab, we ask questions in fundamental neuroscience and use this to guide the development of novel prosthetic decoders. And then we can also use these prosthetic experiments to push back and ask questions about fundamental neuroscience, how the brain incorporates the device into its repertoire of movements. And then finally, we take what's learned from these prosthetic experiments and then translate that to our sister lab, the Neural Prosthetics Translational Lab, where we have a human patient as part of an FDA phase 1 clinical trial termed BrainGate 2. This is a 50-year-old individual who has ALS, or Lou Gehrig's disease, where her peripheral nervous system is degenerating slowly over the course of about 8 to 10 years. And she's left with little ability to control her limbs except for a little bit of movement in some fingers. But she received the cortical implant in December of 2012 and has been able to use that to use a computer and type out emails, etc. And is so far has achieved some of the highest performance in terms of human neural prosthetics to date. And this is really exciting and stuff that's ramping up now. So now I'll show you. This is from some of the colleagues in my lab. This is an experiment with a rhesus macaque monkey who is controlling the white cursor. The green targets are placed on the screen, and the monkey has to modulate its neural activity, which roughly corresponds to thinking about moving his arm. But the position of the cursor is controlled exclusively by the neural activity. So he's able to do this relatively fluidly and rapidly. If you can hear this audio, there's a little bit of a click in the background, and that's the monkey's receiving a juice reward every time he touches a target successfully. So monkeys love to work for juice, and they'll do this for many trials in a row. So now you can imagine moving to a keyboard type task where we have more targets corresponding to letters on the keyboard. And in this case, we're going to light up individual letters to have the monkey type out a specific message. Now, of course, the monkey we haven't taught the monkey to write. He has not acquired human language, and if we were able to do that, we would be in the wrong field. But if he makes a mistake, he has to go and hit the backspace keys. This is real time, and he's able to achieve typing rates which are on the range of what you can type, with a thumb on certainly one of the older cell phones. So, yes, this monkey is named Jenkins, and he's very capable. So this is very encouraging. This is, but how do we know that this actually translates into that the design of this prosthetic will actually work in a human subject? So our colleagues who developed this particular algorithm termed the refit Kalman filter. If you're familiar with Kalman filters from applications in robotics and aerospace engineering, it's a sort of fundamental algorithm that allowed us to land on the moon. It's also what we use for neural prosthetics, but tailoring it to the specific application is where all the secret sauce lies. So the question is whether these very highly customized algorithms will then work in human subjects. So our other colleagues working with subject Q, the 50-year-old human patient, tried virtually the same algorithm design and showed that she was able to successfully move a cursor on a screen as well. And there's some newer data which will be unveiled at the Society for Neuroscience conference showing some very exciting, very high performance control. And so if you stay tuned in the next few months, you'll see some even higher performance communication prosthetics in action. So I've told you about a number of the challenges and limitations that we need to address as a community before we can push these truly into the clinic and have this as a technology that people can just purchase. So most of my research, I should mention that I'm here representing the work of many individuals in my lab. Most of my research is focused on trying to understand the basic motor neuroscience and how the brain actually works. And so these questions we want to address are how the brain uses sensory feedback, how it plans and controls movements, and how it's solving these computationally demanding problems to control overactuated systems in your body. And this is something that's actually very challenging to program a humanoid robot to solve some of the problems that your body solves, that your brain solves trivially every day. So to do that, I work with rhesus macaque monkeys, which you've seen already, and their bodies are similar enough to the human body that their brains are solving a very, very similar computational problem to control the muscles. And their motor cortex is homologous to the human motor cortex. And so as it turns out, what the recordings that we make there tend to look very similar to what we expect to see in humans. So this is their motor cortex. It's scaled down in size, but it's solving roughly similar problems. And what we'd like to do is to record from every single neuron in this region in real time. Now, we can't necessarily do that, but the goal is to scale up to as many as we possibly can. So if we take a cross-section through the motor cortex and look at the architecture, we see a very characteristic layered structure, which was first discovered in histological studies back in the early 1900s. And the architecture of the cortex here has been extensively studied anatomically. So Ramon Cajal, through stains and histological preparations, was able to start the process where we can figure out the microarchitecture of the cortical layers. And so what we found is something summarized by this wiring diagram, essentially, and this is really the statistical connectivity patterns between different layers in the cortex. But this doesn't tell a full story. This is all sort of a static map that's untied to the actual functionality. And this is our best guess is what we think different cell types and different layers are responsible for. But so far, we have not had any techniques which allow us to tie the functional recordings to these static anatomical maps in the same pieces of tissue. And that's a pretty fundamental limitation. What we'd like to do is use very highly detailed anatomical information and then tie that to functional recordings. And that's one of the major pushes, one of the major motivations for Obama's Brain Initiative, which is a large national effort undertaken by the National Science Foundation, National Institutes of Health, and DARPA. And they are looking at, they're still trying to figure out all that's encompassed under this sort of national project, but the idea is to fund the development of new tools, techniques, methods, and that will allow us to scale up the types of questions that we can ask in neural systems. So right now, our electrode arrays correspond to maybe listening to one of these cells in the local vicinity, but not really seeing the entire picture. So we want to change that. So the current technology, as I showed you before, is really sparsely sampling. For every neuron that we record, we're probably ignoring between 10 and 100,000 neurons that are in the local vicinity. So we're listening to a very, very sparse sampling of the neurons through some swath of motor cortex. And our goal is to actually image every single one. We want to see at single cellular resolution the live activity of those neurons. So how might that work? So first I'll show you, this is a movie of what we call calcium imaging in a mouse's visual cortex. So this is a movie by Jim Marshall, one of our collaborators at Stanford, and he has presented moving bar stimulus to mice and then recorded this population of neurons as the mouse is viewing the stimulus. So I'll show you this. Show you this once more. You can see that individual neurons are flashing, and that they're flashing in response to as the neurons spike, their fluorescence changes, and we can actually image that directly. And then what we can do afterwards is analyze the pixel values, and in this particular case, we can look at the orientation of the bars, which cause those neurons to fire preferentially. So this is sort of proof of concept experiment showing that they can back out some of the properties that we know about the visual system using this imaging modality. So, there are many ways of recording neural activity in the brain. You probably have heard of many of these. We can record intracellularly through a technique called patch clamping. We can perform extracellular recordings, such as those which I've been describing. We can also use ECOG or EEG or MEG. And all of these techniques exist at some spatial scale, whether it's down to microns or up to millimeters or meters. And these all give us the ability to record temporal resolution down into milliseconds or sub-milliseconds all the way out to hours. And those are incredibly powerful. But there's this big gap in the middle. So there's other tools like fMRI, which have neither the spatial nor the temporal resolution, but give us access to the entire brain and are completely non-invasive. So we can functionally localize specific things using human subjects and cleverly designed experiments. And that's incredibly powerful, but it's not really going to give us the... It's powerful for certain types of questions, but for us, where we really want to understand the microarchitecture and how that leads to function, we're probably not going to get it out of that. So the last thing I'll mention is that there's other imaging modalities that traverse these spatial scales, but only at very slow temporal resolution. So what we want is to address this giant hole in the middle. And that's calcium imaging, which is not quite, does not achieve the temporal resolution of the others, but traverses this wide spatial scale and has enough temporal resolution in the sub 100 millisecond range to give us very useful data and hopefully address the limitations. So how does this calcium imaging work? I've shown you this teaser video, but I'll go through and describe the process and some of the equipment that we're building to get this technique working in primates. So back in the 60s, several scientists discovered through studies of jellyfish, there was a specific protein which fluoresces when you excite it with a specific wavelength of light. So more recently, we've worked out the structure of this protein, and it has this very characteristic and beautiful beta barrel structure. And what this is doing, this barrel structure is... when we cut this away, we see that there's a fluorophore, which is a small molecule hidden inside the center of this barrel. And this is what absorbs a photon and then absorbs an excitation photon, and then which kicks an electron up into a higher energy level, and then this electron relaxes back down and emits a photon in this slightly different wavelength. So the entire structure surrounding that fluorophore exists purely to protect it from the solvent, the water around it, which would otherwise quench the fluorescence and sap that energy away. So the excitation photon comes in. If there's any water around that molecule, it'll quench it and prevent it from fluorescing. So this is great. This is an incredibly powerful tool. And we can use that to statically image individual neurons. This is a neuron that's cultured, and we can use antibodies which have this GFP, the green fluorescent protein, attached to it to label specific parts of to label the specific parts of the cell, and then we can build up an image that way. But this is static. How can we? This isn't going to show us the activity of this neuron. This is just going to give the static image. So, what can we then do to make this a sensor for the activity of the cell? So, if you actually look at what happens when a neuron is spiking, there's a very complex set of mechanical and chemical processes at the level of individual proteins, and it's highly complex. Now, we are accustomed to thinking about the wide voltage swings as the neuron spikes, and these voltage swings are due to the movement of ions from one side of the cell membrane to the other. There's potassium, sodium, chloride, and also calcium. And if you look at the activity of calcium ions, it is very highly correlated with the overall spiking activity of the cell. So if we can detect the level of intracellular calcium, we can use this as a marker for the actual spiking activity in the cell. And some true dark geniuses in a number of labs around the world. Some of the most prolific ones are at Genelia, the Howard Hughes Medical Research Campus outside of DC. They have plugged together proteins that exist elsewhere in nature to alter the fluorescence properties of this green fluorescent protein based on the concentration of intracellular calcium. So this is a protein called calmodulin, which they have combined with M13, which actually comes from the myosin motor protein. And what happens here is this M13, they've removed one of the beta sheets that one of these green barrel stays, if you will, that protects that fluorophore in the center. And when calcium is present, these four orange, there's one hidden, but these orange spheres correspond to individual calcium ions. When those calcium ions are present, this calmodulin changes its conformation, acts like a hinge, and actually swings this M13 domain into place, closing up the barrel and allowing it to fluoresce. So when the cell's spiking, all of a sudden this thing closes up, can fluoresce, and that's where you see the bright flash. And then as soon as the intracellular calcium level drops again, it opens back up and no longer fluoresces. So here's a rendering of that in a little bit more detail, just to give you some physical intuition. You see this sort of large hole in the side of the beta barrel, and then this blue alpha helix will close into that gap when calcium is present. So this is actually an incredibly powerful, because this is purely genetic. This is a protein which every neuron and pretty much every cell in your body can produce this if we can find a way to get that DNA coding for that protein into those cells. And so we use that with viral delivery, where we actually take viruses such as AAV, the adeno-associated virus, and strip out the payload which causes the virus to self-replicate, and instead put in segments of DNA that code for these specific proteins. Now, they will then you can genetically target subclasses of neurons, or you can target all the neurons if you'd like, and have those viruses then deliver these chunks of DNA inside such that the cell's own machinery will begin producing these proteins for you, and fill the cytoplasm with this, what we call GCAMP6M in this case. And once you've waited long enough for the neurons to produce this protein, usually on the order of several weeks, then you can start imaging this. But how do we actually image this? Before I get there, when we build these movies, we again want to then treat this in a rigorous quantitative framework. So to build the sort of analog traces that correspond to the underlying state variables of those neurons, in this case, we just average the pixel values within a region of the video corresponding to a specific neuron. And that gives us this black trace here corresponds to the average pixel value of this neuron over the course of time. And the gray trace underneath is an intracellular recording. It's an electrode that's been placed inside the cell. And you can see these sharp deflections are the action potentials, which correspond very closely to the sharp vertical deflections in the calcium trace. So you'll notice that it responds very rapidly. As the cell spikes, you can see immediate changes in the fluorescence of the cell, and then you have a relatively slower decay. And this slow time constant of the decay could be problematic, but what we can see is that we can actually, just by eye, you can go back and pretty much recover individual spikes, and there are algorithms to do that, and a number of people are working on this right now. It's an active area of research. But we can see fundamentally there's nothing stopping us from understanding the firing patterns of the cell from this video imaging. So the next question is, okay, what hardware do we need? How can we actually image the fluorescence in vivo in live tissue? And we use a technique called two-photon scanning laser microscopy, where we have a laser. In this case, it's an ultra-fast femtosecond pulsed laser that we send through two rotating mirrors called galvanometers. We scan this back and forth across the sample. And at any point in time, the laser is focused just on one area of the tissue, exciting the fluorescence there. This then goes back through these rotating mirrors, hitting our detector, photomultiplier tube in this case. So we sweep this back and forth over time, and we can actually do this extremely rapidly using state-of-the-art resonant galvanometers to achieve frame rates of about 30 hertz or even higher if we limit our field of view. So you can do this quite rapidly, and there are trade-offs between how much tissue you're looking at and how fast you can revisit those areas. So photon fluorescence is, I'll talk just a little bit about this phenomenon and then and why we use it. In the single photon fluorescence case, where we're exciting directly with the wavelength that is absorbed by the fluorophore, we have a lot of scattered light that's out of the focal plane of the microscope. So in this case, we have this yellow line corresponds to the focal plane, and all of this excitation light coming in from the top is scattered off of the scattering medium. In our case, it will be neural tissue, which is highly scattering. And that contributes to background and reduces your signal-to-noise ratio. So there's a technique called two-photon fluorescence, where we use excitation light at double the wavelength, and we require two photons to hit the fluorophore at exactly the same time. This is statistically highly improbable, so you need incredibly high powers of light, and so that's why we use very, very powerful lasers that pulse for very short periods of time, so that the time average power is very low. And when we do that, we can get a very precise focal volume, which has a number of advantages, allowing us to increase at greater depth due to less scattering of the excitation photons. We can use higher peak excitation powers, and we get lower background and higher SNR as a result. And so I recognize this is a bit of a whirlwind tour of this, but if you're interested in any of this, please feel free to follow up with me afterwards. So just to illustrate this in the same framework, in this case, you absorb a photon, excite your electron, and then jump back down, re-emit a photon. Here we're re-emitting the same fluorescence photon, but we require two red-shifted photons of double the wavelength, half the energy, to simultaneously hit this and excite one electron to jump up. So this works already in flies and in rodents. What do we need to do to get it working in primates? So this is all of this work began really only a handful of number of months ago. So we're up and running. We a few days before I got out here, we got this microscope working for the first time. And so I'll be talking about our anticipated challenges and not actually presenting the final product. So if you check in with us in the winter, we will be doing this in primates for the first time then. So this will be very exciting in the next few months. The key anticipated challenges that we're addressing in the design phase of this to adapt this imaging modality to primates are we need physical access to optically image the brain. That's actually remarkably challenging. We also need to stabilize the brain tissue. Since we're imaging at micron resolution, really, we need to keep the brain tissue from moving more than a few microns. And third, we need to process and store incredibly large data sets. We're generating on the order of 4 terabytes of data a day, and this greatly scales up the both storage and processing requirements from our traditional electrophysiology experiments. So I'll tell you a little bit about what we're doing with that. So to address the first concern, just physical access to this tissue, we've designed a titanium chronic implantable window, which will allow us to bring an objective lens right up to the surface of the brain. We're working with human neurosurgeons, but I get to do most of this design with some undergrad mentees of mine. And this is designed to, this titanium bone likes to grab onto titanium. This is something that we can implant and that will comfortably remain there long-term, hermetically sealed. This is something that we can leave for upwards of years. And one of the key design considerations was to be able to change the silicone window in here. So if we do injections through this window, we can then remove it, replace it, and clean this as a sterile operating room procedure. But we really wanted to retain that flexibility. And what you'll notice is this giant blue thing in this cross-sectional view is the objective lens that we're using for this microscope, which is about 3.5 centimeters across. So this is a huge, huge lens. And being able to bring this to within 2 millimeters of a monkey's brain is actually remarkably challenging. So much so that we had one of our students had to write a MATLAB collision detection and design optimization algorithm to optimize the geometry of this to fit around the objective lens while also still working with the geometry of the skull. So what we thought was going to be a relatively straightforward design challenge turned out to be relatively complex. We have our first prototypes of these manufactured. We're testing these now, and the first implants will go in January of this year. So we also needed to figure out how to mold those silicone hermetically sealed windows. So we've actually, working with others who have designed similar products for different application areas, have designed this mold. We needed to figure out a way to make this optically next to perfect and block as little light as possible. So that's something where despite my background in electrical engineering, having spent several hundred hours in the machine shop here at Thayer, actually was an invaluable experience as we go through and do a bunch of this mechanical design and then physically building things by hand. So this is the final product. You can see it. You can kind of see it on the front of a smartphone, and the goal is to make it completely optically transparent, not block any light. And we're iterating on the design to further improve that. But so far, it looks like we have something that will actually work. So this is quite exciting. Second, we need to design a microscope that will work in vivo with primates. So working with a company called Bruker, formerly Prairie Microscope Systems, we have developed, my colleague Dan O'Shea is showing this here, we have developed a in vivo microscope which features 6 degree of freedom robotic control. This is, it moves in X, Y, Z. It also has a 2 orbital degrees of freedom, which I'll show you on the next slide, as well as a resonance scanner on the Z-axis for the objective lens. So this is incredibly important to allow us to get precisely target specific regions in the brain which don't correspond to bringing the lens in on a vertical axis. So this sounds relatively mundane. You need to image from an arbitrary geometry. But this is actually something that's very, very hard to find, and we had to work with these scope manufacturers to develop these custom parts specifically for this application. So once we set this up, we will probably be the only lab in the world with this capability. But we, of course, hope to, by doing this with a industrial partner, they will sell this and then disseminate it to the broader neuroscience community. So our hope is that by doing this, we can do this design effort once and then have it benefit the whole community. So we also have 2 independent laser paths, as well as a third through our resonant galvanometer, so we can actually photostimulate at the same time that we're imaging. This will be important down the road for certain other classes of experiments that I won't have time to talk to you about today. And then we feature a number of different light paths for to give us a lot of flexibility to try. This is very much an experimental tool, so we want as much flexibility to play with the optics down the road as we can possibly get. And we first got this working Friday of last week, so this is now up and installed and running, and I can't wait to get back home because we're just starting to play with this. So this is this 2-axis orbital nose piece, the Generation 3 variant of this. The customizations which we're designing into the next version of this will be direct servo control of these orbital degrees of freedom, as well as a light guide coming out of the back, which collects all of the non-ballistic the both ballistic and non-ballistic scattered light to greatly increase the number of photons that we can collect and send to our PMTs. So this will be extremely important for imaging deep in the cortex, where a lot of the emitted fluorescence in the tissue is actually lost due to scattering in the. So we're trying to recover every single possible photon that we can, and going to great lengths to do that. So the second major challenge is stabilizing the brain tissue. As the monkey with pulse and respiration, you actually see that the brain tissue moves on the order of sometimes millimeters within the brain. So we need to apply gentle mechanical pressure, and we've designed a device which fits inside our imaging cylinder and provides subtle mechanical pressure to stabilize the tissue without actually interfering with the basic biology there. So finally, I'll just talk about storing and processing large data sets. And then so after this, we'll open it up for questions. So our storage requirements are very large. One experiment produces anywhere between 1 and 4 terabytes of data per day. This requires redundant storage, and then the computational challenges while working with this data are also very high. It can take upwards of 15 to 20 hours just to process some of these data on a traditional workstation, depending on what analysis you want to run. And so we need alternate ways of doing that. And so what we're starting to work with is distributed cluster computing using tools such as Hadoop and Spark, and a package called Thunder produced by one of our collaborators, Jeremy Freeman at Janelia. And these are ways of these are essentially implementations of Google's MapReduce algorithm for very simply working with large clusters of machines in a resilient, distributed way, such that you can run an analysis, have that transparently, but very much under the hood, farmed out to a large cluster of machines, then those data collected back, or the resulting data collected back together and presented to you. So this can decrease your processing time from 16, 20 hours down to the order of 60 seconds or so. Oftentimes you get a scale up of about 3 orders of magnitude in terms of speed, and of course it's dependent on what analysis you're running, but these three tools all together are starting to become widely used in data science in general, particularly in other areas of Silicon Valley, and this is something that we're going to be pushing on pretty heavily in the next year. So, and I should mention also that we can run this on Amazon Web Services or local clusters. Increasingly, it's more and more attractive to just run this out on somewhere in the cloud and get the results when they come back. So our goal kind of in summary here, our goal is to image populations of hundreds or thousands of neurons simultaneously in vivo in awake and behaving monkeys. We want to be able to do this deep, anywhere between, from the surface down into about, upwards of 0.8 millimeters or a millimeter into tissue. This is a fundamental limitation of optical imaging. The brain is opaque, but we believe that we can, with two-photon imaging, we can get a usefully deep distance into the cortex. And then we also want to identify individual neurons across experimental sessions day to day, and this kind of greatly expands the possibilities for what types of experiments that we could ask, where we look at learning experiments that go across multiple days. And if successful, this will enable us to address many new classes of experiments, connect structure with function in living tissue, and hopefully yield new insights into cortical processing mechanisms and how the basic biology gives rise to these very complex behaviors that we see, just human capabilities in general. So, in summary, I've gone through talked about the operational principles of neural prosthetics, the challenges and limitations facing these, and then use this to build out why we need more tools and more basic fundamental neuroscience knowledge. And then I've told you, of course, about the calcium imaging work that we're developing in primates. So with that, I would like to just highlight the opportunity that exists in this field for engineers such as yourselves. This is the building that I work in, the Clark Center. It's quite nice. We're always looking for help, people with background in computer science, electrical engineering, robotics, machine learning, EE, mechanical engineering. So these are very, very highly sought after skills and can be incredibly helpful. So if you're interested in any of these aspects of what I talked about today, just feel free to send me an email or reach out, and I'd be happy to talk to you not just about opportunities where we are, but also elsewhere around the country. There's a lot of research in this area in many places. So with that, I will take questions. Yeah.
Well, you apparently do come from Silicon Valley because you called your silicone silicon.
So I mentioned both silicon and silicone. We're working with silicone for the imaging windows and silicon for the implantable arrays. Is that? Did I swap that? All right.
Silicone, which you called silicon.
Well, my apologies. I'll I see.
I have a question. What kind of resolution, optical resolution are you expecting?
Yeah, so we can essentially get down to diffraction-limited spot size if we want to. So people use the calcium imaging technique to image down to the level of individual spines on neurons, which are...
I'd like a number for the optics you've designed.
Oh, for the optics that we've designed, we can get down to 0.8, 0.7 microns. So the bodies of the cells that you see there, that's not our major goal. So the bodies of the cells that you see there are anywhere between 20 and 30 microns. So if we're primarily interested in that somatic calcium signal from the body of the neuron, we actually would rather have field of view than cellular resolution.
That would be my next question.
Yeah.
What was your field of view?
Our field of view, it depends on the objective lenses that we're using, but we're designing around, for a 16x objective, we can hit about 750 microns square. And with a 25x objective, it gets down to about 350 microns. And then we're looking at potentially working with manufacturers to develop custom optics that would allow us to then hit wider fields of view, where we could again sacrifice resolution for increasing the number of cells that we hit.
So asked another way, what are your resolvable spots?
Uh,
How many resolvable spots?
So typically it's like 512 by 512 is a typical within your field of view. You'll have that type of resolution. Now it's completely arbitrary, but you're trading off a certain number of spots that you can acquire per second. So you're trading off the resolution versus the frame rate. So with your galvanometers, you can park that laser in as many places as you want, but when you do that, you can't go as fast. So typically what people use is either 256 by 256 or 512 by 512, and with resonant galvanometers, you can hit frame rates of about upwards of 30 hertz. But oftentimes people use standard galvos and get frame rates more like 4 hertz in systems where the neural dynamics aren't changing fast enough to require that. In our case, the monkeys are making very rapid reaching movements, and we want that very fast dynamics.
Yeah, maybe the answer is too soon
Yeah.
for this answer, but is the cortex more like an CAN bus, or are hardwired channels to certain areas?
We don't know the answer to that question. We think of the so motor learning over the course of weeks will probably really establish new neural connections, and there's certainly a lot of flexibility in the network. But it's probably more static connections than a very a flexible fabric where the activity can modify the connectivity in on the time scale of seconds and minutes. But that's a great question that a lot of people are going after right now, and maybe different in different areas of the brain. Think of architecture more. The more flexible architectures may be up in the prefrontal cortex, and then in the basic motor system, where it doesn't necessarily need to change on very rapid timescales. It may actually have a slightly different approach.
Is there any hope that one could monitor the activities of a fairly large number of neurons without having to make a hole in the skull, either through electrical sensing or through other means?
Yeah.
What's the state of the art in that direction?
Right. So the state of the art in terms of non-invasive neural recordings, we can use MEG and EEG. People are looking at other potentially interesting and exotic ways. There's certainly things like ECOG, which don't actually go underneath the dura, but underneath the into the cortex, but are still sitting on the surface of the brain. But as far as I know, there's there's of course PET and fMRI. But we don't have anything that will give anywhere close to cellular resolution, and those are fundamental physical constraints. The analogy of, if you want, it would be like, trying to listen to an individual violin in an orchestra while standing outside the building or something. It's very, there's a lot that we can learn from those imaging modalities, but if you really want, high temporal and spatial resolution, it's almost certainly not something that we can do in humans. Now, there are other techniques such as molecular ticker taping, where we can look at potentially recording the activity of every single neuron in the brain in something like a mouse, where you can then extract the brain at the end of an experiment. So this is actually using DNA polymerase and creating DNA strands which act as a recording of the neural activity over time. So the electrical activity of the neuron changes, adds defects to this polymerization reaction in the DNA. This is still a pretty far out technology, something that people have just started to talk about, but is potentially exciting for mouse and smaller organism experiments.
I follow up, if I may. Is it necessary for some applications, like for example, a person that does not have control over their limbs, to control a robotic arm? Is it necessary to have that level of detail that you get from individual cell monitoring, rather than just monitoring a whole area of the brain and kind of getting the
Right. So you can certainly do some low bandwidth control using EEGs or using other, but something that we think of as being clinically useful that'll work in a real world environment. Something like EEG can get the signal will be completely swamped from the electrical potential introduced by just even movement, your muscle activity, electrical noise, some of these things. So there are many in the community who are working on non-invasive ways for designing low bandwidth prosthetics. I don't think a full arm or a rapid communication prosthetic that will achieve typing rates that are closer to what we think of as natural typing. It'll be much harder to achieve that with a non-invasive. Yeah.
I have a question on sensory feedback.
Yeah.
The original prosthetic hand couldn't close on the object.
Right.
So her mind could operate it, but it didn't sense the object.
Yeah, absolutely.
Now there's
Yeah.
signal.
Yeah.
You have to image the signal. But
Right.
My question is, how does the feedback signal incorporate with the activation signal?
Right. So that's a fantastic question, and actually that was when I started my Ph.D. 3 years ago. That was what I was principally interested in addressing with the design of these prosthetics. So there are people who are doing research in exactly that. So you instead of just visual feedback, you want proprioceptive, tactile feedback. You want to you want to increase the ability for this prosthetic to use the other senses that we already have. And so some of our collaborators at UCSF, the Flip Sabi's lab, has done some truly incredible work where they implant the same microelectrode arrays in the somatosensory cortex. So this is just behind the primary motor cortex. On the other side of the central sulcus, you have the somatosensory cortex, which is responsible for aggregating this, the somatic signals from your sensory neurons. And we have the same map of the body, but shifted back to this other location corresponding to this incoming sensory input. And what they've done has been to implant this electrode array into the somatosensory cortex and then electrically stimulate with unique patterns of activity corresponding to different target locations for this monkey who's trained to make reaching movements. And they're actually able to do this with enough fidelity that they just by the patterns of stimulation on the array, they have monkeys that can reach to targets that they can't see. So they stimulate with a pattern, telling the monkey where it should be reaching, and then the monkey will successfully reach that target. So that's indicating that they can send in a lot of information into the brain highly successfully. And when they actually, even more interestingly, they find that they can that the monkey optimally integrates that cue with visual information. So it's quite interesting. What I didn't get to tell you about too, yeah. Yeah.
And then we better stop. It sounds as if you're saying that the feedback may stimulate other neurons. And I was wondering, does it reincorporate itself, modify the activation signal, or does it activate separate neurons, and then how do those communicate?
That's exactly the types of questions that we want to answer, and we don't currently have an answer to. So we know that there's sensory and motor areas are not as functionally distinct as classically thought. They're very highly integrated, but we don't know how, say, proprioceptive information is encoded. We know generally where it's coming in, but we don't know exactly, what the processing steps are and the exact connectivity patterns that translate that from that input into guiding and controlling a movement. And it's our, belief and hope that we can answer those questions and then use that in obvious ways to inform the next generation of prosthetics. So,
If I may
yeah.
to this, I can see that you can go on.
Yeah.
What I will do is suggest to anyone who's interested in following up with any questions,
Certainly.
speak with Eric right now, or to contact him later. Thank you very much, Eric.
Thank you. Thanks, everyone.